and principles of bioinformatics with a few examples. So the aims are the introduction to the course. I'm going to outline the structure and the content of the course. I'll then go through an overview of what bioinformatics actually is and how it's used in sort of modern day biology. We'll then cover sort of brief essentials of molecular biology, including the central dogma of molecular biology and DNA, RNA, protein sequence, how they're stored, and we'll just cover briefly about homology as well, which will become important later on. We will also cover some various case studies, which I will show you in a bit. So briefly, just to say sort of who I am, I'm currently doing a PhD in archaeal genetics, and they are prokaryotic, so like bacteria, but they were considered, emphasis on were, an separate domain of life, along with the eukaryotes and the bacteria. So you can actually see that up here represented, and archaea were considered a sister group to the eukarya or the eukaryotes. Now, this model is now out of date, and actually the current thinking is that the eukaryotes should branch out from about here in the archaeal tree. So whether there are 2 or 3 domains of life is sort of still debatable, but it's more like 2. Many archaea are extremophiles, so they'll survive in extreme conditions. You can see this down at the bottom. So in sort of deep salt lakes, in hypothermal vents, things like that. Interestingly, they're also why flamingos are pink, if you're interested. It's because the plankton and the krill that the flamingos eat are themselves pink because of the pigments found in archaea within these organisms. So they're found in hot springs, as I said, deep sea vents, salt lakes, and obviously in guts of eukaryotes such as ourselves and cows. They do also live in normal habitats though, but DNA replication in archaea is very similar to that of eukaryotes, hence the interest for my research on DNA repair systems. And of course, importantly, there are no known pathogens, so happy days. I'll go through this in the session. Essentially, it's just saying that there are some quite complex ideas that are going to be presented to you over the next few weeks doing this course. And you know, I do want you to understand the information that I'm presenting to you. So if you don't understand anything, there are a few things you can do about it. The first thing is what you're sort of listening to now. There are video recordings of sort of each lecture, if you like. And I'll make these available to you via Teams. However, please do email me with questions. I will give you my email address or post them on Teams as well. I'm happy to do that. If you have a question, please do ask it. So as an overview to the programme, this is what's going to sort of be what you're going to be working through over the coming weeks. So you can see that here. We've got an introductory session followed by a session dedicated to molecular biology to give you a kind of grounding in the sort of academic theory that you need to understand these bioinformatic principles. We'll then cover a separate lecture, of which there are 3, introducing you to various bioinformatic techniques, databases, systems, etc. And after each particular lecture on a bioinformatic technique, you will have then a computer practical where you can actually have a go at using those techniques and systems. And as you can see, there are 3. So one is on databases and something called blast sequencing. The next one, multiple sequence alignments and protein domains. And the final one is genomic and protein information. So I think it's sort of prudent to begin by introducing what bioinformatics actually is. And I think this definition is sort of quite a nice one. There are many other ones out there, but I think this one suits our purposes best. So it's defined as a collection, classification and storage, and analysis of biochemical and biological information using computers, especially as applied to molecular genetics. So essentially using computers, software, computer packages in order to analyze biological and biochemical data, generally in the field of genetics more than anything else, and biochemistry, but there are exceptions as well. And hopefully by the end of this talk, you can come back to this particular question and hopefully go through each of these bullet points and identify whether they are examples of bioinformatic techniques. So, why bioinformatics? Well, it's hugely important in biological research in the 21st century. We have a wealth of biological data in various databases, and actually it's really important. It's used on a regular basis during my doctoral work, but also actually just in science generally. There's probably not a day goes by that most people don't use some form of bioinformatics. So as I said, it's used in pretty much all areas of biology, but especially genetics, molecular biology, that sort of area. It allows for acquisition of knowledge to sort of direct experimental procedures and form hypotheses. So that's quite important. Again, you will either use bioinformatics at the start to direct your experimental procedure and your hypotheses. You might use bioinformatics during the actual collection of data, or indeed to analyze data afterwards. So it really is an integral part of modern biological research. And it's faster, which I think is a sort of rather understated pro, if you like, of bioinformatics, is because it's computer-based, it is far faster than typical sort of archetypical wet bench studies in the lab. So you only need your computer and your brain, essentially. So bioinformatics kind of varies from sort of small scale sort of individual lab sort of projects. So things sort of, individual experiments for, a research project that you might do, for example, at university, undergraduate level. And these can be simple searches to find information about genes, proteins, look at what the proteins look like, look for mutations and what impact they may have, all of that kind of thing. They can also be very large projects and actually can be a single person analysing a wealth of data, vast quantities of data, potentially up to sort of hundreds of thousands of data points, or indeed very large multi-lab projects. So this can be a global collaboration between labs. So there's really two scales, and generally this course will be focusing on the small scale type of analyses. So why should you care? Hopefully you've sort of come across this already in kind of what I've said, but essentially it's a really important tool in modern biology. It forms a grounding for most biological degrees and indeed careers. And it's important for you in terms of transferable skills to be able to understand how to work with big and large data sets and interrogate and sort of inform your decision making via these techniques. It's highly sought after by many employers in many career areas, and actually if you are looking at doing any form of degree in the biological sciences, whether that be the kind of typical science route or indeed medicine and that kind of thing, so biology, biochemistry, it's actually a really important skill in many career areas and degrees. So actually generally now bioinformatics is considered a more sort of transferable and more important skill than indeed experience on the bench doing sort of science in test tubes. So I thought I'd give you a couple of examples of bioinformatics in action, specifically relating to the COVID pandemic. And this one, essentially what this particular sort of paper tried to do was to look at the different variants that have been introduced into the COVID genomes compared to strains already in circulation. So again, hopefully you're aware that this particular coronavirus, SARS-CoV-2, wasn't a kind of new thing. Coronaviruses have been around for years. Things like MERS or indeed the initial SARS coronavirus a few years ago. So all they did was they wanted to see what mutations does this particular variant of coronavirus have that the other ones perhaps didn't, that may point them in the direction of why it was so serious and highly transmissible. So what they did was they took the genome from COVID, the new strain, so SARS-CoV-2, and the genome looks like this. Don't worry too much about what these mean. It just stands for open reading frame. You'll come across that later. There are all these genes in the genome. They were particularly interested in looking at the S or spike gene. And so what they did was they essentially sequenced a load of variants of these strains and looked specifically at the spike protein. So the protein looks like this. So they sequence these particular proteins, and you can see the novel coronavirus isolates, one from Wuhan, the initial one, and one from the USA. So you can see the two here compared to all the others which were known in existence at that time. It's not an extensive list, but it's a few. And what they could do was something called a multiple sequence alignment. So they take these protein sequences, which you'll see later. Proteins can be abbreviated in terms of sequence to their individual amino acid residues, and each amino acid is given a single letter code. But we'll get to that later. And what you can see is you can align all these sequences together, so they sort of match up, if you like, and then you can spot the differences. And hopefully you can see that there's a PRRA sort of set of sequences of amino acids that are present in the novel coronaviruses, but not in the previous ones that were known to us. And that's part of the spike protein. So potentially the PRRA sort of motif, if you like, could contribute, in theory, to the degrees of severity and transmissibility of the novel coronavirus. The other example, just sort of quickly, was that this other paper decided they wanted to find out how do the co-circulating sort of strains of COVID vary and persist within populations. Essentially, they sequenced genomes from loads of coronavirus isolates between March and June of 2020, so quite early on during the pandemic, and they wanted to know or find out how long different lineages last in the population, and indeed how many independent introductions of these COVID isolates into the UK occur, and how many are actually distinct, and how many die and are not retained in the population, and by extension, how many actually are retained in the population. So if we looked at the data, again, this is a bioinformatical procedure that allowed them to do this, and you'll see why later on. Essentially, the purple X's here mark the differentiation, if you like, from a non-UK sort of purple case into a UK isolate. So they sort of indicate importation events from outside the UK into the UK. And you can see this from this graph. So what they found is that sort of 1,200 independently introduced genetically distinct chains or strains of COVID were indeed introduced into the UK. And they also looked over time. So if you look on the left-hand side here, you can see that sort of very early on in sort of March, April, May, etc., they were detecting lots of new lineages. And actually, as we progress into sort of April, May, that very much sort of died off, in that actually there were less importation events during that time, most likely because of that lockdown. Interestingly, lots of these sort of lineages were not observed for more than 4 weeks. So you can see that with this graph here. So as time went on, so from about April, you can see that more and more of these isolates actually died off after 4 weeks. So that's sort of quite interesting. And if you're interested and wanted to read, I'll provide a link to that particular paper on the Teams channel. So hopefully that gives you an idea of what bioinformatics can do or has done recently. And again, if you're interested, both of those papers that I've just talked about, and indeed applications against or for vaccine development are here as well. I'll put them in the Teams channel. So in terms of biological data then, what you're going to be looking at over the next few weeks are sequences, first of all, so of nucleic acids and proteins. We're then going to look at mutations and polymorphisms within those sequences. And then we'll sort of transgress into looking at protein structure and function for the final session. But this is just to show you that bioinformatics can do all these other things as well. And indeed, this is not an extensive list, so it can do many more things than this. So I'm just going to talk about sequence data for a little bit. So nucleic acids and proteins can all be represented essentially as linear strings of information. This is sort of quite good for you because it's fairly cheap to acquire. So DNA, RNA, and indeed protein sequencing is fairly sort of rudimentary in most labs now. And computationally, they're easy to handle and manipulate through computers. So this links into something called the central dogma of molecular biology, which I will briefly explain now, but it will become more apparent next session. Essentially, if we start with DNA here, and don't forget it's a double helix containing anti-parallel strands. This sort of duplex of DNA can be replicated and can give rise to obviously more DNA duplexes. However, through the process called transcription, we can actually read certain parts of this DNA called genes, and we can transcribe them or sort of make a molecule or a strand of RNA that corresponds to that particular gene, kind of like as a sort of copy of a text or something. That's quite important. You know, you make a copy of it, and then you can do with that copy what you will. It's not sort of quite as important as the raw material up here. And that RNA can then be translated via the ribosome, don't forget, in vivo, into strings of amino acids linked together that then fold up to form a polypeptide or a protein. So when protein synthesis occurs, this is just to hopefully remind you that essentially we have this reaction going on up here, where we have the joining of this amino acid and this amino acid via the carboxyl group of one amino acid and the amino group of another. They will join together via a condensation reaction. So don't forget we are liberating water here. And they join together to form the peptide bond, and therefore in this case a dipeptide, but this will continue to form a polypeptide. It's done by the ribosome, which you can see down at the bottom right here, and that is kind of done, if you like, by reading the genetic code, which you can see here outlined in this table. I'm not going to cover this too much now, I'll cover it next session, but essentially you have certain codons in your mRNA sequence. So let's say you have a CAU, that will actually code for a histidine amino acid, and then so on and so forth. So just get used to using this table, but we'll see it later on down the line. So just to introduce you to the structure of genes and sort of vaguely what they look like, we'll look at a eukaryotic one first. So don't worry too much about the detail. All of this detail will become apparent next session. Essentially, the start of the gene, you have what's called a promoter region. This is a bit of sort of DNA that allows the start site of the gene to be detected, and it marks the start of the gene. At the very end of the gene, you have the kind of equivalent of that, which is a terminator region, which again marks the end of the gene. So when we are going to transcribe this gene during the process of transcription, the enzyme that does this knows where to start. So it's RNA polymerase, knows where to start. It will read all the way through, and then it will stop reading at the terminator. So it's a kind of start and stop signal. Don't worry about the UTRs for now, that will become apparent. But essentially, although we start transcription at what we call the +1 site, so right essentially by the promoter, the actual coding sequence, the actual sort of amino acid coding codons, if you like, don't really begin until sort of further on where the first exon is, but I'll get to that later. Either way, you have something called an untranslated region. So this will be transcribed, this sort of grey area, but it won't be translated. More detail to follow, just accept that for now. Eukaryotic genes are interesting because they are interrupted. The coding regions, which are called exons in red here, are interrupted by intron regions. Essentially, these are non-coding parts of DNA that are spliced out later on, and again, you'll see that next session. They're removed via a process called splicing, and all these exons will then join together to form one large red chunk, which then can be read by the ribosome. And again, the ribosome will stop reading at the end of the final exon, and again, you have an untranslated region towards the other end of the gene. Just worth a note at this point, however, that it's computationally quite challenging to take a DNA sequence such as this for a gene, including the introns, and actually predict where the intron-exon boundaries actually are. It's quite tricky to do that, and I will talk more about that later on as to how we might get around that problem, but it's quite challenging to do that. So if you have a DNA sequence like this, it's actually not particularly useful to you in its current state. So it's worth just pointing that out now. Prokaryotic organisms have a slightly different structure to their genes, and I'm not going to talk too much about it because actually we're going to focus on the eukaryotic systems for this course. But again, they have a promoter region and a terminator region. They've got UTRs, so the same principle there. However, their genes, the main difference is that there are no introns. Okay, they do not exist in prokaryotes generally. There are, as with everything in biology, there are exceptions, but take it as read, they don't exist. And essentially each gene is sort of backed onto each other, but they're clustered in structures called operons. And essentially all this is it's a collection of genes, doesn't have to be 3, but a lot of the time it is, that have similar functions. They may all work together, for example, in tryptophan, which is an amino acid, metabolism in bacteria. And that particular operon can therefore be turned on. We can transcribe it, we can make protein out of it very quickly, but actually we don't have to do that for each individual gene to make tryptophan. If we need to make tryptophan, we can just turn on this one sort of promoter, if you like, and all of these genes will be expressed simultaneously. And what that means is we can have very efficient ways of regulating genes and turning on biochemical pathways rather quickly. But as I said, we're not going to worry too much about operons. We're actually going to focus predominantly on eukaryotic genes throughout the course. So, just to point out now that it's fairly easy to do that translation on a computer if you know where the start and stop signals are in your mRNA molecule that has been spliced. Okay, so it's not with introns present. Once these introns have been removed, if you know where the start of this exon is and the end of this exon is, you can, in theory, use the genetic code to translate that fairly easily. But as I said, it's more difficult to predict the gene model. So, for example, knowing where the introns and exons are is frankly quite a challenging process. There are ways to get around it, which is actually to combine the genomic data of the genes with the transcript data from sort of mRNA sequences. So you would compare the two essentially, and the mRNA that has had the introns removed would tell you where those introns are in the gene model. But don't worry too much about that. Just have an appreciation for the fact that if you did want to translate in silico a eukaryotic gene, you need to use spliced mRNA. So essentially without introns, and you need to know where the start and the stop codons are. Even though once you've done that on a computer, let's say you've translated it, so you have a protein sequence of amino acids, it is still, I would say, verging on impossible to sort of predict accurately how that protein will fold in vivo. So protein folding can't yet be accurately predicted. Again, there's slight caveats to that, which I'll talk about during the protein session, but we still can't do that. We cannot take a sequence of amino acids or even a gene sequence and accurately predict what that protein will look like. So talking about protein function then, essentially because technology has evolved so much over the last sort of 10 years, our knowledge of genes and the amount of gene sequences out there has not really, well, we've got so many that actually technological advances have meant that we have far many more sequences and genes than we do proteins, essentially, and indeed knowing what those proteins do. So you'll notice that more than 50% of the proteins, and there are a lot, so 215 million, in this particular database, have the words unknown or hypothetical in their title, meaning it's potentially a protein, and this is what the protein might look like, but we have no idea what it does or even if it's actually translated. So we actually then need to have experimental data to prove that a particular gene sequence in that database actually does encode a protein, and actually that protein does a particular job. But all this is very time consuming and financially quite costly. So there are lots of sequences, as you will see, in the databases, but actually most of them, we don't know what they do as of yet. As I mentioned, we cannot predict protein function or indeed structure very easily at all from first principles, but we can therefore do something to overcome this, which is by looking for homology, which I'll talk about now. So what is homology? Essentially, homology means descent from a common ancestor. So those two, let's say, sequences or proteins are evolutionarily related to each other. They have descended from a common ancestor. And that is really important. If genes or proteins are homologous to each other, we can therefore say that they have descended from a common ancestor, and they are likely, again, important, it's not sort of guaranteed, but they are likely to have either the same or similar functions. So that's important. So essentially, if you have a new protein, you don't know what it does, the easiest way to go about finding that is to sort of look for any similar proteins that are potentially homologous to that protein, and that might give you clues as to what this novel protein might do. Again, homologous genes will have descended from common ancestors, and so they'll show significant similarities in their DNA or basal sequences. Note similarity, not identity. So it doesn't mean that they're going to be 100% identical. It just means they are similar. Because don't forget, evolution will have changed that. So essentially, the takeaway for this is that we can infer homology between genes and proteins based on how how similar their sequences are. So we can compare the sequences, see how similar they are. If they are very similar, we can infer that those might be homologous sequences. Note the phrasing, they might be homologous. We need to do some more checks to see whether indeed they are. So just talking about what homology is not, it's not the same as similarity. So I mentioned this earlier. Homology is binary. Things are either homologous or they are not, because they are either descended from a common ancestor or they are not. So even if you have very similar things, they may not actually be homologous because they might not have evolved from a common ancestor. And it's not about having the same name either. So things such as the cyclin-dependent kinase 1, which is an enzyme, in Homo sapiens is a homologue to CDC28 in yeast and CDC2 in S. pombe, again, another form of yeast. So these things are homologous to each other. They just have different names because they have been discovered independently in different organisms. And again, CDC2 in pombe encodes this cyclin-dependent kinase, but in Cerevisiae, CDC2, again, same name, actually is not at all a homologue because it's frankly a completely different protein. It's a catalytic subunit of a DNA polymerase called DNA polymerase delta. Don't worry about that too much. It's involved in DNA replication. So just be aware of that. It's also not an analogy. So analogous sequences are formed, or proteins, are formed from convergent evolution. So essentially, two independent lineages have sort of evolved similar features and characteristics by chance, but that's not the result of descent from a common ancestor. So examples are body plans we have placental mammals here and Australian marsupials. Again, body plans are very similar, but they are not actually evolutionarily related to each other. The other example is between a dolphin at the bottom here and an ichthyosaur at the top. So again, the dolphin is not a descendant from the ichthyosaur. They have evolved millions of years apart and from sort of separate lineages. So they are not actually in any way related to each other. So they're not homologous at all. They are analogous because they have very similar features morphologically, but actually evolved separately. So how can we use sequence data? Well, just to sort of clarify on this, we can, as I mentioned, identify homologous sequences. So we can look in databases and we can search by number, name, or species. And we can look to see if there are any potentially homologous sequences out there compared to your query. Or we can take those sequences and we can align them together. The key thing is that these sequences have to be homologous to each other. There is no point aligning sequences that are not related to each other. However, if you do have sequences that are related to each other, you can line them up essentially side by side, as we saw with the COVID example earlier, and we can compare them. And that's often the basis for lots of sort of analysis further on. So an example of a sequence alignment I will just show you now is something called sickle cell anemia, which hopefully you have heard of before. It's an inherited blood disorder. It's caused by a homozygous mutation in the beta subunit of hemoglobin. It's a glutamic acid to valine substitution, which is an amino acid. And you can see that normal red blood cells that don't have this, typical sort of biconcave disc. But actually, if you have sickle cell, it actually causes distortion of your red blood cells and therefore reduced oxygen carrying capacity and actually quite severe anemia. So how do we know this? Well, what you can do is you can take sort of normal human variants at the top here. So we've taken 4 variants and you can see their amino acid sequences as a sort of string along here. And you can see they're all pretty much the same. However, you can see the 2 sequences from sickle cell patients. You can see this stark difference here, where there's a E, which is glutamic acid, at position 6 in the beta globin chain here for the normal variants or the kind of wild type variants. But the disease alleles give rise to a valine at position 6 in sickle cell, and that is indeed the mutation that occurs. Now, you can see that there are other variants. So, for example, here we have methionines present for these two sequences of these two variants, and indeed lysines for the variants 1 and 2. But as you can hopefully imagine, that clearly doesn't make any difference to the disease state. Okay, so it has no impact on the disease having sickle cell. But hopefully you can sort of see that aligning sequences in this way can tell you quite a lot of information quite quickly. Again, don't worry about the details of this, that will come later. You can also align sequences and compare them and use them to construct phylogenetic trees. So essentially sort of evolutionary trees. And this is what was used. So they sequenced ribosomal RNA just because it's sort of fairly ubiquitous amongst organisms and quite easy to get hold of. There's lots of it. They aligned them and in this paper by Carl Woese allowed them to infer that actually there are 3 separate domains of life: the bacteria, the archaea, and the eukaryotes. But as I told you earlier, that's the current thinking. This was the 1970s. The current thinking is that the eukaryotes actually belong from within the archaea. So it is changing, but hopefully you can appreciate that by comparing sequences of different organisms and indeed species, we can conduct analysis to create a phylogenetic tree. I briefly want to talk about genomics. So again, this is a bioinformatical procedure whereby we can sequence genomes and analyze those sequences. And as you can see here, that in the 1970s, we were able to sequence 170,000 base pairs, which was a virus, T2 phage. And actually, as the years have gone on, we've been able to sort of sequence more and more genomes and larger genomes. So we have actually the human genome, so 3.2 billion base pairs. And actually, the largest genome known currently is this particular plant, 150 billion, which is quite astounding. It's not actually been sequenced as of yet, but I'm sure by the end of this course it may well have been. But this is just to show you that actually, our sort of computational abilities has increased substantially over the years. We've been able to sequence these organisms and sort of annotate them so we can now use them. And you'll be using the human sequence later on. Interestingly, the cost of sequencing, if you were to look at this, in sort of 2004, etc., it cost between sort of 10 million and 100 million pounds for a genome. But actually, as the time has gone on, technology has increased more than Moore's law. If you're into computer science, then you may wish to look this up. Essentially, it's a prediction of the cost of sort of technology because of technology advancing. But because of this, now, back in 2019, sequencing your genome actually costs sort of in the region of a high-end computer. So actually, and this will continue to fall. So actually, it's pretty rudimentary stuff now, and you can via 23andMe and that kind of thing get your genome sequenced fairly cheaply. Just to talk about the UK Biobank, essentially this is a collection of approximately 500,000 individuals. Again, this is from 2018. And essentially it's a whole array of different biochemical and genetic markers from sequencing that allows you to store this information and relate it to disease phenotypes predominantly through various genomic approaches. So you may have heard of the 100,000 Genomes Project. It's actually surpassed this now, but it was to sequence 100,000 genomes from patients. Again, mass bioinformatical procedure to analyse that data and to essentially look for disease variants and see whether any particular variants of genes can correlate with phenotypic outcomes in the form of disease. So again, this just shows you the kind of vast quantity of data that's now out there and available. The final example I want to talk about is that of protein modelling. So bioinformatics has been used on COVID, as you can see here, on the spike protein especially, to model this computationally as a structure. How they did this we'll, again, touch on later on. And you, in fact, will be looking at these structures as well. But you can see it produces quite nice images, quite nice pretty pictures. And again, you can actually use bioinformatics software to change the image and how you see it. So this is a ribbon diagram here. This is a wire diagram. And you can see all these structures in all of these things. And again, here you can look for sort of binding of different things to the protein. But I'll cover that later on. Just have this in your head now that this is another use of bioinformatics. And again, you can zoom in and even on the angstrom level, you can really look at interactions, hydrogen bonding here between side chains of amino acids, between a protein ligand and the spike receptor. And you can model that. It's 3.4 angstrom distance, and you can do various other things as well. You can also, via molecular dynamics, model this spike protein, as this paper did, via, again, computer simulations and molecular dynamics. So what they wanted to do was identify on the spike protein where would be good to target vaccines to, because actually the spike protein is covered in loads of green, as you can see here, carbohydrates and kind of glycoproteins. So what they wanted to do was model potential access sites for vaccine development. And they did this, and they were able to create this really nice sort of video, if you like, of this molecular dynamic situation. So you can see all the green carbohydrates and how they're moving around on these spike proteins embedded into the membrane. But again, this is high-end bioinformatics that is allowing this to happen, and they then created the vaccine as a result of that, targeting free areas of the spike protein that are not covered by carbohydrates. And that is covered in this paper here, if you are interested. Again, link on the team's channel. But sequencing is only part of the job. So all genetic sequence are just mixtures of A's, T's, G's and C's for DNA. It's all well and good having that sequence, but what you need to do is essentially overlay annotation on top of it. I.e., you need to identify where the genes start and stop, what are genes, what are not, what are part of introns. You need to identify promoters and terminators, and actually give each gene specific identification markers so that other people can search data on the same gene. Annotation of genomes works in two fashions. The first is automatic, which is done by computers, which is very fast, but as a result makes quite a lot of errors. Or manual, which is done by humans actually using knowledge from research to actually manually annotate genomes, which takes a long time, but is far more accurate. And again, we'll talk about that later on. So this is just showing the example. If you were to sequence something, you might get an output like this, just A's, T's, G's and C's. But the question is, where are the genes? Is this part of one gene? Is it part of two genes? Where's the promoters? Where do the genes start and stop? Are there introns in here or exons? We don't know. And what does that gene do? So again, annotation is important. It's all well and good sequencing, but you need to then overlay on top of this the information that you need. So just to wrap up, the aims for the rest of the course are to understand the uses, the importance of bioinformatics tools in research, to recall concepts behind these things I talked about, which is what you'll be doing. So database searching, similarity searching for homologous sequences and proteins, and therefore inferring whether things are potentially homologs. To conduct multiple sequence alignments, which we saw today. Again, you'll be doing that and looking for differences, etc. And to model protein structure via three-dimensional modeling software, which again, you will also be doing. So you need to hopefully gain experience in these bioinformatic tools and interpret the output intelligently. It's quite academically rigorous, as I mentioned, so please do engage. It will push you academically, I'm sure, but that's hopefully the point of it. And hopefully you come away with a far better understanding for your A-level course, certainly for the A-level scientists and biologists amongst you, to allow maybe the context of things like protein structure and genes and transcripts and transcription and translation, all of this stuff to hopefully sort of come together a bit more logically. Please do ask for help. So if you are stuck during the computer practicals or even during the other sessions, if you're not sure about something, please do just say. Either ask me at the end of the session, email me, or put it on Teams. I'm happy to make time outside of the timetabled lessons to go through things with you, because essentially there's no point you doing this and not understanding it, right? I really do want you to understand what it is you're doing and hopefully enjoy it. Hopefully you'll obviously enjoy the course as well. Please do bring your laptops with you. I've put that there at the bottom. You will need your surfaces, and you will be required to download software prior to the computer practicals. But again, I will give you the details of that. It's all on the team site and in the course handout. I will give you details of that in due course. So that's it from me for this particular introduction session. Hopefully you have an appreciation for what bioinformatics is and how it's used and sort of what you're going to be doing over the next few weeks. Again, don't worry too much about the detail for now. That will come later on.